Well, thank you so much for inviting me. And, I want to say hello to all my friends that I wish I was there with you. I want to say hi to Mike McDermott, a fellow Canadian, and want to say thank you to Brent and to Dr. Adler, who I've known for a long time. So, John, one of the things I admire most about you is your iconoclastic approach to things, and this meeting and what you're trying to do is no exception to that. So what I'd like to speak about today is talk about focused ultrasound and maybe draw some parallels between ultrasound and radiosurgery and where each of these fields can learn from each other. So it turns out that I'm going to be talking about three things. One is making lesions, two is stimulating, and three is opening the blood-brain barrier with ultrasound. And I think that there are some parallels with radiosurgery here as well. Now, the history of focused ultrasound and radiosurgery are actually quite similar in that they started about 70 years ago. And initially, focused ultrasound was too bulky. You had to open the skull because you couldn't get the ultrasound through. And really, radiosurgery took off, and really to make lesions in the brain was much more advanced. Over the course of time, though, focused ultrasound has caught up. And I would say that in the realm of stimulation, that focused ultrasound is actually ahead of radiosurgery. So we'll talk more about that later. The current focused ultrasound systems to make lesions have about 1,000 beams that are focused onto a single spot in the brain. And of course, the problem is the penetration through the skull. This is a problem that we do not have with radiosurgery, of course. It looks something like this, where the patients are fixed in a frame and the entire assembly is put in the MR machine. And then you can aim at any spot within the isocenter of the beams and make a thermal lesion by applying energy. And it usually takes about 13 seconds or so to apply the energy. And you do this sequentially, gradually increasing the energy delivered and the temperature. You can measure the temperature in real time using MR thermography. And so what you do is you basically build an onion shell, one shell at a time, and you keep on adding extra layers of this onion until you make a lesion and until you get a clinical effect. In the case of tremor, you of course monitor the tremor, and you can keep on shooting and making the lesion bigger and bigger until the tremor goes away. So the advantage of this, of course, is that you have monitoring of the physiology in terms of the patient's clinical status, and you also have monitoring of the temperature and the size of the lesion. These lesions look identical to radiofrequency lesions. So here is a focused ultrasound lesion after 1 day, after 7 days, after a month, or after 3 months. And they look, you cannot tell the difference between a thermal lesion made by ultrasound and one made by radiofrequency. Now, it's now been 6 years. We published this paper in New England, a randomized trial of focused ultrasound for thalamonomy for tremor. And what was interesting about this is that it was quite effective, but this is not without side effects. And the adverse effects are seen in this table. And you'll see that, for example, the most common side effects are paresthesias, about 38% acutely. And this goes down to about 14% at 1 year. The second most common side effect is a gait disturbance. You'll see that about 36% of patients have an acute gait disturbance, and at a year, still 10% of them have a gait disturbance. There's also things like contralateral weakness, hemiparesis, et cetera. So the issue is that this has side effects, and I would say that the side effects of focused ultrasound thalamonomy are probably higher than the adverse effects seen with radiosurgery thalamonomy. And we can speculate why this may be. But we've been interested in understanding why the patients have side effects. And what we've done is we've mapped out all the lesions that had side effects. And what we found is that, in fact, some of the lesions encroach on the medial meniscus, and these lesions give you paresthesias or sensory side effects. There are some lesions that encroach on the internal capsule, and then you get a hemiparesis or weakness with these lesions. And then for the gait disturbance, we found that the lesions that extend below the thalamus in the dentatorubothalamic tract in the subthalamic area are the ones that are most closely related to the gait adverse effects. So we're trying to better understand the relationship between lesion location and size, the efficacy, and the adverse effects. And in this way, we can better avoid these adverse effects while maintaining the benefits of these lesions. Now, in Toronto, we actually have all 4 modalities to treat tremor. So we have radiofrequency lesioning, of course, we have DBS, we have focused ultrasound and radiosurgery. And we offer all of these to the patients with tremor. And we give them this table and we tell them it's like going to the restaurant and you have to choose what you'd like to have on the menu. And we feel equipoise in that we feel that all 4 of these therapeutic modalities are of similar value, of similar efficacy, and have similar profile of adverse effects. So we feel quite comfortable in giving the patients the choice of which one of these 4 treatment modalities they would like. And in the patients with only tremor as their main problem, with essential tremor, for example, over 90% of them are choosing focused ultrasound. So they see it as non-invasive. They see it as being something that, they call it a laser operation, most of them, and the results are immediate. And the majority of them are voting with their feet and asking for the focused ultrasound. Interestingly, our referrals for tremor have increased tremendously after the focused ultrasound. And we've had, for example, 200 patients on the waiting list for tremor assessment, and the majority of those will not have focused ultrasound. Many of them will have actually DBS or other things. So when people come through the focused ultrasound route, they often end up with another diagnosis or another treatment. So it's interesting that the focused ultrasound has really raised the level of the water, and all ships have gone up with the tide. Now, you can imagine that if you treat tremor in the thalamus with focused ultrasound, that there may be other disorders that might also be amenable to focused ultrasound lesion treatment. And indeed, there's a whole slew of them, including Parkinson's disease, tumor ablation, depression, pain, epilepsy, and so on, that could also be treated with focused ultrasound. And I'll just show you some examples of these. So here, for example, is palidotomy. So you can do palidotomies with focused ultrasound, and there is soon going to be published a multi-center trial of this. You can, of course, make lesions in the Palidotothalaamic tract, and Daniel Jean-Monod, this is his favorite target to treat the bradykinesia and the tremor in patients with Parkinson's disease. You can also do lesions in the subthalamus, the subthalamonomy, and this is the group from Madrid, Jose Obeso, that does this type of surgery. You can also do bilateral thermal capsulotomies for the treatment of obsessive compulsive disorder, and Jinwoo Chang from Korea and his group have done this. And again, Daniel Jean-Monod has done central lateral thalamonomies to treat pain. And so you can see that there are a number of lesions that can be done with focused ultrasound for therapeutic benefit in both movement disorders and in the psychiatric realm. Let's now go away from lesioning towards stimulation, and let's talk about stimulation. For stimulation, we're not using this big helmet with 1,024 sources, but rather we're using a single handheld source of ultrasound, which is applied to the skull through the hair, as is seen here in the middle panel. So the fact that you can stimulate the brain with focused ultrasound is pretty clear. And I'm going to show you this video from an animal experiment done by Jamie Tyler. And basically, this is a rat that has focused ultrasound being delivered at 1 hertz. And you can see very clearly that the focused ultrasound is producing a very robust effect in the corticospinal tract. So it's very clear that focused ultrasound can stimulate the brain. We've written this paper reviewing the mechanisms through which focused ultrasound could stimulate the brain, and we published it in Mark Georges' journal. I think Mark Georges is one of the next speakers. And basically, we don't know how it works, but the mechanisms through which stimulation, focused ultrasound stimulates the brain is poorly understood. But there are a few main hypotheses. So, for example, we know that there are mechanosensitive ion channels embedded in the neural membrane. They're shown here, for example, and any minor distortion of the membrane might change the properties of these channels, inducing, excitability to the neural tissue. We also know that there can be temperature changes with ultrasound. There can be bubble formation and cavitation and mechanical forces, and all of these could lead to excitability of neural tissue. So these are some of the main leading hypotheses as to how focused ultrasound may work to excite neural tissue. There have also been reports of direct stimulation of the cortex using focused ultrasound, producing, for example, sensations, paresthesias, when focused ultrasound is applied over the somatosensory cortex. This is a group by Yu in Korea. There's also been the same group producing phosphenes by stimulating the visual cortex. So it is quite clear that a single transducer of ultrasound through the skull can stimulate the cortex and produce physiologic effects as are seen here. In fact, we've summarized all the human and animal studies, and you'll see that many areas of the brain have now been stimulated in humans with focused ultrasound, including motor cortex, sensory cortex, the caudate, the frontal cortex, the thalamus, and the visual cortex. So all of these brain areas are now have been stimulated with focused ultrasound in humans, and this is a rapidly increasing field. In our own group, we've been able to show that one can influence the excitability of the motor cortex by applying focused ultrasound. And although we cannot get a motor contraction directly using focused ultrasound, what we can show is that the sensitivity to magnetic stimulation can be altered, and there can be excitation of the motor cortex if you pre-treat with focused ultrasound. So in this particular experiment, we do transcranial focused ultrasound and beta burst, and then we apply a TMS coil. And what we have shown is that if you apply a focused ultrasound train and you wait even an hour later or 2 hours later, you can see an increased excitability of the motor cortex. And this is shown here in this graph, where you can see that the magnitude of the evoked potential increases after focused ultrasound. So it's quite clear that in humans, you can use focused ultrasound to increase the excitability of the brain, in this case, to increase the excitability of the motor cortex. This paper was published about 2 weeks ago in Annals, if anyone is interested. We've looked at looking at various elements of dosing of the motor cortex, and interestingly, we've also looked at the penetration of focused ultrasound through the brain. And our models show in cadavers and in vitro models show that, in fact, the focused ultrasound can be focused at a depth about 8 centimeters, as far as 8 centimeters from the surface of the brain. And what this means is that it is now possible, at least theoretically possible, to stimulate the basal ganglia, even in the subthalamic nucleus. So in the future, we may be able to use focused ultrasound for cerebral mapping, for example, of motor, sensory, speech functions. And we might be able to also use focused ultrasound to study the effects, to mimic the effects of DBS at deep targets, for example, the STN or DBS. And one can even speculate that if you applied focused ultrasound on a more chronic basis, that you may be able to mimic the effects of DBS and do DBS without DBS, but rather with ultrasound. The last topic I want to cover is the possibility of using ultrasound to open the blood-brain barrier. And this is done not by ultrasound itself, but by the co-injection of microbubbles. And the microbubbles will expand and contract with each ultrasound wave, and they will in fact shear open the tight junctions between the endothelial cells as shown in the video, and they will then, this will lead to a destruction of the blood-brain barrier. And once you disrupt the blood-brain barrier, then the floodgates are open and all kinds of interesting things can get into the brain. So this is shown first in Toronto by Todd Mainprice and his colleagues. And here is an example of a patient with a brain tumor, and they've shot 8 different shots of focused ultrasound, 9 rather, a 3x3 matrix, and then they give gadolinium, and they see that in fact the gadolinium extravasates through the focused ultrasound treated area. And this gadolinium extravasates is maintained for about 24 hours. So the blood-brain barrier reseals after 24 hours. So you have a 24-hour window of time through which you may be able to get some molecules into the brain that otherwise would not. And I'll remind you that the blood-brain barrier keeps everything out that is 400 molecular weight or higher, so that this means that you can now start thinking of getting things into the brain like proteins, like genes, like oligonucleotides, like antibodies, etc. And as a proof of principle, I'll show you this picture here on the left, where this is an injection of a gene called D-tomato directly into the brain of a rat. And you can see that when you inject this gene into the brain, it expresses and the brain becomes red. You can do the same thing by applying focused ultrasound and giving the virus intravenously. So here in this example, the blood-brain barrier has been open. There's been an intravenous injection of this virus, and the virus has gone and penetrated the brain only where the blood-brain barrier has been open. And in this way, you can get gene therapy without opening the head. So you just open the blood-brain barrier, you inject the virus, and then it goes selectively to the area that's open. And this then is a very exciting new way of thinking of doing gene therapy. There are some downsides because when you inject it into the brain, you need a very small dose. You need 1,000 times more viral vector when you do it intravenously. And of course, the virus would get in not only in the brain, but would go all over the place in the body. So it still needs some adjustments until this is ready for prime time. So in conclusion then, we talked about focused ultrasound to make lesions. We've said that focused ultrasound can be used to stimulate. I think that when it comes to lesions, the Gamma knife is way ahead. But I think that when it comes to stimulation, I think that the ultrasound is way ahead and moving rapidly. The opening of the blood-brain barrier is also a very exciting new possibility for focused ultrasound. And I'll finish off with this saying, I think that I think the take-home message today is that, but to paraphrase Nietzsche, what does not kill me makes me stronger. I think if we paraphrase that, sublethal radiosurgery produces neuromodulation is a paraphrase of that. Namely, that radiosurgery can be used to alter neural elements and could alter their excitability and produce neuromodulation. And with that, I want to thank you all, and I want to thank all my colleagues, and thanks very much for the invitation.
Thank you, Dr. Lozano. We have just a few minutes for questions. Any questions?
Andres, how long does the excitability of cortex last after FUS?
Well, so there's an acute excitability, but there's also plasticity. And so we now know that after theta burst, if we do theta burst stimulation for 5 minutes, we can get effects hours and hours later. So we let people go home after a while, but we want to bring them back now the next day or the next week and to see whether there is a persistent and enduring effect. Mark George will speak about TMS, and there too, there are long-term plastic changes that occur as a consequence of a relatively brief treatment of the brain. So we think that there's going to be really long-lasting effects of focused ultrasound. We don't know how long, and we don't know whether we will have to treat repeatedly or whether this is going to be an infrequent dosing that will be required to produce a long-lasting change in excitability of the brain.
Andres, Mike McDermott, any comments when we're talking about adenovirus and DNA viruses about the difference between blood-brain barrier disruption and intravenous versus intra-arterial delivery of the virus?
Yeah, so the viruses are huge, as you know, Mike, and yet when you open the blood-brain barrier, they can sneak through and get in. The advantage of intra-arterial is that you could reduce the dose quite significantly. I think if you give it intravenously, then it's going to go throughout the systemic circulation.
Right.
So I think that there's an advantage of thinking of intra-arterial delivery of these agents to reduce the dose and reduce the toxicity and to limit the spread throughout the body. And one can almost think that you would like to infuse through the cerebral arterial system and then pick up and filter out or dialyze out the virus, from the veins,
Yeah.
so that they don't reach the rest of the body.
Yep. Thank you.
One more.
Thank you. Very interesting talk. A quick question. Let's say, do you think that a system like this can be put in a fixed geometry in certain points in a machine like ZAP to activate, the bubbles? I mean, how important is directionality and those type of things?
So how important are the bubbles
Meaning, the ultrasound, how many, suppose I want to put this in a fixed geometry inside of a ZAP machine, to use it when needed. I mean, how
Right.
important is directionality to get the bubbles, to have the effect?
Yeah, so people have used a single source with bubbles and ultrasound to treat brain tumors, to open the blood-brain barrier in the vicinity of brain tumors. And so there, it seems to be that a single source is sufficient and you can focus it. So there's some focusing properties and directing properties. So that is sufficient to get a focal opening of the blood-brain barrier in the context of enhancing the delivery of chemotherapeutic agents in the context of brain tumors.